Hello everyone. Welcome back to another event of Aquatic and Fishery Science Talks. And this is AFS talk number 48. For those who are new, let me introduce who we are. I'm Deepak. I work as a fisheries scientist in Australia. I'm hosting this webinar today. And with me on board is Dr. Sajana from Central Inland Fisheries Research Institute in Barakpur. And the two other board members, we're still waiting for them to join in. One is Mukta Menon from Seema Farai and Dr. Pradesh from Texas State University, USA. Our board members will be serving as co-host for this webinar. And if you have any technical issues, please get in touch with them. A little bit of introduction about AFS Talks. Aquatic and Fishery Science Talks is an ongoing webinar series, and we try to do this every 2 weeks. We invite speakers around the world to share information and developments in aquatic science. Our aim is to bring science to the public by linking students, professionals, researchers, farmers, and other hobbyists. So this platform is open to everyone who loves hearing a science talk and also giving a science talk. Though most of the talks are science-based talks, but today we have a talk which is more in line to giving guidance for like career guidance. And it's very rare we get a few people who can actually give the guidance how to take your career on from a fisheries background. In AFS Talks, we have a big team of volunteers, and they work as coordinators, distributing, circulating our posters and letting people know about the upcoming talks. But anytime, please feel free to jump into www.afstalks.org, and all the links are available in the website. And there we have a Facebook, Instagram, LinkedIn profile, and everywhere we post our upcoming talks. And we have a WhatsApp group, and there are 3 groups. And if you want to keep updated about the talks, please join in one of our WhatsApp groups. The rules for the webinar or webinars are free to attend, but you would need a profile name for your Zoom account. Please note, we don't issue any e-certificates for attending any of our webinars. Please keep your mic turned off all the time, unless the host requests you to turn on and speak. If you have any questions during the talk, please post them in the chat box so everyone can read and understand your question, and for the speaker himself. And we will give you a chance to talk directly to the speaker. The speaker has 45 minutes for the talk and another 30 to 40 minutes for interacting with the audience. We give more emphasis on the discussion because we want to serve, for the reason what you are here. Please turn off your webcam while the speaker delivers the talk, but it will be great to see you in action when we have the discussion after the talk. Let me introduce the speaker of the day. Welcome, Dr. Manush. Manush is an alumnus of College of Fisheries, Panangad, and has a PhD in stress physiology from CIFE. And further, he has postdoctoral experience through JSPS in Japan, Uniform Services University, and University of Pittsburgh in USA. He has research experience in the preclinical drug discovery stages to mitigate myocardial infarction, metabolic syndrome, and cancer. Manush also has recently graduated Executive MBA in Healthcare from the Cassie School of Business at the University of Pittsburgh. Manush is currently a research faculty at the University of Pittsburgh, and this talk is going to focus on the biomedical opportunities for fisheries graduates in the international arena. Manush will share the challenges during each stages of his career and how they shaped his thinking process during the career transition. So welcome again, Dr. Manush. The floor is yours now. Thank you.
Thank you, Deepak. I'm audible to you, right?
Yeah.
So, good morning, good afternoon, or good evening, whichever time zone you are in. Okay, and thanks for AFS organizers to invite me for giving me this talk. And thank you everyone for joining with me here today. So actually, this is an opportunity to revisit my career journey. So when I'm talking to you. So a couple of weeks ago, when I was chatting with Deepak, we thought this would be a nice topic to discuss. So that's why I'm here. So I am excited to see at least some of the familiar faces, or at least the names here. So hope we can have some discussion after the presentation. So before that, some of the housekeepings, like you can put down the questions in the chat and we can discuss this at the end of the presentation, because it's hard for me to keep up with chats while talking. So today I will talk about my path into biomedical research, a glimpse of my research areas and the challenges I faced. So I hope this would be helpful for somebody who wanted to take this as a career options for you. So I was a part of 1994 batch at Kerala Agriculture University College of Fisheries, Panangad, and finished my master's from CIF in 2001. At that time, actually, I was not having a clear path what I'm going to do. So some of my colleagues left to US for pursuing master's or PhD. So at the end of my master's, I got a PhD admission in CIFE, and Dr. Paul was my PhD mentor. So I was talking to him about completing PhD while I pursue all the different options. So during that time, IPCC, like International Panel for Climatic Change, was coming up with a stunning report about the impact of increasing global, like the temperature and global warming and its impact on different habitats. So I have decided to work on thermal biology, heat stress response in fish models. So we also had collaborative projects with the Department of Atomic Energy, Government of India, to work on thermal biology. And I have to say that Dr. Paul created a nice team environment in the lab, in biochemistry lab. And during my PhD work, our team, the lab team, could get around 13 papers in stress physiology. So I think those papers came in handy when I was applying for international fellowships. So with that, we could network with professors working in Germany and Japan to secure international fellowship. So I applied for DAD fellowship in Germany and JSPS in Japan, and I got invitational fellowship in 2006. So that time, actually, 2 people from India got into JSPS in biology section. So I have to say a couple of things here. So research productivity and how you can relate your research interest to the host institution abroad. So you need to keep in mind while you're preparing this proposal. And this will be key in securing these fellowships. Soon after I left for Japan, like many of my colleagues and juniors from biochemistry lab have secured opportunities abroad in US, Canada, Europe, like it was very successful. So I'm happy that this was a nice trend building up in CIFE. So while I was in Japan, I used my time to learn different molecular techniques using zebrafish model. I was taking the molecular responses of heat stress in Japan for my study. So then I worked in Uniform Services University and in collaboration with the NIH for 2 years in cancer biology. So by 2010, I got an opportunity to work with the developmental biology at University of Pittsburgh using zebrafish model. So since then, I am working in Pitt on different projects like cardiometabolic disorders. We know that COVID hit in 2019, 2020 time. So all the labs got shut down. And during that time, I didn't know what to do with that. So to make use of the time properly, I have joined MBA program in healthcare and graduated in 2022. So I'll come back to my story in detail a little later. So now we look at like what are the opportunities out there which you can go into. So let's talk about if any of you wants to run the same path, what are the options? So here, these are the regular exits from CIFE to other institutes to international careers. Like first of all, completing bachelor's, and like while doing master's, you like pursue the GRE or TOEFL and do another master's outside India. So come into CIFE, pursue a PhD while doing master's in India and in CIFE. Then as the next step, which I have taken that route, like completing bachelor's, master's, and PhD in India, then compete for international fellowships. Then you have, if you know someone, if you can network properly, you can also get direct postdoctoral fellowships in US or any other developed countries. So while I was doing this research in India, like there was very few ARS positions being advertised. Like for 6 years, there was no positions advertised continuously. So this is another route, like you can take, like you finish bachelor's, master's, and PhD and get into a scientist position, and you can apply for sabbatical positions abroad if you have India government funding. Like any of the funding organization in India, if you can get funding, you can easily get sabbatical positions for like 6 months to 1 year to get experience. So there is also another route, like you can go on deputation for international assignments while you're working as scientists. Like there are different ways people go out of CIFE or any other Fisher's Institute. So next I will talk about the JSPS fellowship because I have the experience in going through that route. So I have availed like long-term invitational fellowships for 24 months. So when you apply with your proposal and CV, you will be sending directly to JSP's office, and they will accept the application twice a year, April and September. For standard long-term program, you need to apply through proper channel in Indian institution. At that time, I think it was like HRD office in Delhi was handling that. So there are also short-term programs you can go for research for these like 6 months period maximum. There is another short-term senior program which is given to senior professors and Nobel laureates to do research in Japan in collaboration. They also have a Ronpaku program. I think it is something new to complete PhD research in a Japanese lab as a dissertation work or thesis work you can think about. There are other postdoctoral fellowships also. Now in US, one of the reputed programs is like Fulbright program, and in Germany you have that fellowship that you can apply for. Japan also offers a fellowship for completing PhD. So in this program, you will be admitted to a like Japanese university and registered as a PhD student, and they will be providing fellowships for completing your research. So for master's level, I think Erasmus Mundus scholarship, and somebody has given a talk in detail in some of the previous sections, so you can refer to that. So once you get into biomedical research in US, what are the different options you have? So if you are not entering US as a student visa, the most common route for biomedical research is by securing a postdoctoral position. These are some of the most career options if you if you end up in academic research, like you can see postdocs, research associate, biomedical scientist, or principal investigator, or in a teaching position. These are the different options you have. So once you get the green card, which is a requirement for you to switch into industry, because it's industry people, they don't hire you by giving you green residency or green card. Like while in H1, you cannot switch to another position very easily, especially in industry. So if you like to work for US government, that's another option. There are different federal jobs in the government. And see, I have highlighted here in green, which is like the requirement for all the positions which are listed here in green. The qualification is master's or PhD. But if you have an MBA, so you will also get into the additional roles which are listed here, like program administrator, grant manager, or like a chief financial officer, science consultancy, all the different options. So the fourth quadrant you see here is a startup. It's a different ball game. Your degrees and qualifications does not matter there. If you have a clear vision or idea, and you have the drive to work for that idea for years while it is developing, and you have the ability to convince those investors who can fund you, then this would be a career route you can take. Once you have a successful exit, then you can help invest in developing similar startups so that you can become an investor. So now I will switch gears to give a glimpse of my journey as a biomedical researcher. So then I'll be talking little about my research, and that will give you an idea what you will be doing if you are choosing biomedical research as your profession. So one of the main activity in the field is to reveal the mechanistic insights into the root cause of a disease. And this information will be useful to promote innovation and drug discovery. So this is an overall picture of the drug development process, where a potential drug can make it to a useful medicine in humans. So the regulatory process is very stringent in US. And there are several steps in preclinical and clinical stage. So you can see the percentage of candidate drugs that will become successful. It's less than 10%. And it is a very costly and time-consuming process. And the most expensive being in the clinical trial phase. If you look at early discovery phase, like several thousands of molecules are screened to a lead drug. And one of these molecules tested in healthy patients in clinical stage 1. So that stage it is called like investigational drug. So if it is promising, then it is taken to the next level for mitigating the disease condition in a small set of populations or patients. So this is kind of phase 2. And when it becomes successful, the drug will be tested for efficacy in a large population in multiple centers, multiple countries. Once it is cleared, FDA will review and approve them. And then this drug molecule can be promoted by the company as a useful new drug in the US or like even globally. Oops. Okay. So when you think about preclinical testing of drugs, there are several animal models which are commonly being used. So you can use like two-dimensional cells like stable cell lines, or primitive model systems like C. elegans, Drosophila, zebrafish, to name a few. So these are actually useful high throughput screen models to test the drugs, which means that more compounds can be screened for at a lesser cost. So that is required in the early stages. Some of the drugs which are promising in this stage will be taken into preclinical trials in mouse and monkey models. And this could be used to validate the lead compounds before getting into humans. This is because the physiological and genetic similarity to humans is like, it is the main factor which is considered at that stage. So because this is mostly a fish audience, I will describe how a fish model can be useful for chemical genetic screens. So before that, I will tell you about one of the signaling pathways I was interested in. FGF signaling and its role in development and disease. So why it is important? Because optimal FGF signaling is required for early embryonic patterning, heart development, and other physiological functions. So what happens if FGF signaling is insufficient during development? FGF receptor mutations can lead to craniosynostosis, which means it's like enlarged skull development in babies. So this is normally corrected by surgical methods. So what happens if it is too much? Upregulated FGF signaling in adult humans can lead to cancer. Because this is a very critical pathway, this is regulated by a set of feedback regulators. DUSP6 is a feedback regulator of FGF signaling. And you might be familiar with early developmental stages of zebrafish. You can see here on the right panel on the top. And by using zebrafish, you can visualize FGF signaling in the early stages. And you can modulate those signaling by different techniques like morpholinos, or you can make knockouts. You can overexpress that protein in the zebrafish. So that way you can study them. Then there will be like if it is a protein which is very dynamic, which means that it comes up and go away very fast. So you may need to create a model which is living model so that you can visualize them properly. So how you visualize these signals in a living embryo? So if FGF signaling is regulated by DUSP6, and we can generate this transgenic zebrafish fusing DUSP6 promoter to GFP, then we can see the dynamic changes of the FGF signaling during development. Oh, sorry. So that's what you see here. Like the GFP expression, you can see them on the head region, especially on the mid-hindbrain boundary and rhombomeres. You can see them. So the next step is how you can adapt this into a chemical screen. Once you have the transgenic embryos, you can fill it into 96 well plates and treat them with a new library of compounds, which are unknown compounds. We don't know what the functions of those compounds are. You can fill it like the middle wells, 80 wells on the middle 96 well plate. And the top and bottom lanes, you can have negative and positive controls. Then you can read the fluorescent signals and in an image scanner, and then you can quantify them. On the top right panel, you can see that while treating a compound called BCI, you can see increase in the FGF signaling. So now we have the molecule. Next step is to refine the molecule into a better medicinal, like to improve the medicinal value of that drug. How you do that? You can take up this compound and treat them in increasing doses and see when they become toxic. Then we can think about how to modify the structure of the drug. So look at different regions. So here, for example, unsaturated ketone ring may be undesirable, may be highly reactive, so it can cause toxicity. So that is an assumption you can make. So then you can test it. So when you replace the ketone group with an OH group in the region, so the compound completely loses its activity. So this means that ketone ring is very critical for its FGF activity. So you cannot mess around with that region. So when you work with the different analogs, you can find new molecule. So here you can see BCI 215 is less toxic than the parent compound. So adding a bromine can help in retaining the FGF activity while reducing the embryonic toxicity, which means that BCI 215 is a better drug than BCI. So this is an example how you improve the drug before taking it to mouse or any other clinical trials. So how you use fish model for understanding heart development, because that's the lab which I was working on. So their focus is mainly on that. So the humans have a 4-chambered heart, but zebrafish has 2 chambers. And the blood flow is unidirectional, which means like it is going in a single direction. But you have all the comparable features of human heart in zebrafish embryo. This will help us to study zebrafish and relate it to humans. Also, the zebrafish is transparent during embryonic stage. So it is easy to visualize the development and the beating pattern of zebrafish heart, actually. So here is a transgenic fish embryo which I made in the lab with red blood cells moving through the green chambers. So using this model, you can ask several questions that affects blood flow through the heart. So there is another, like when you work in that environment, you will have to do multiple projects to see what are the potential in each of these projects. So here is another way to study heart looping defects using zebrafish. So why it is important? So externally, actually, human body is bilaterally symmetrical. It means that left and right, you have left and right hands are similarly, like it develops similar manner. There won't be any difference. But internally, the organs are efficiently packaged to function it properly. In majority of the cases, we have normal arrangement of the organs. Like the heart is always towards the left. Then you have multiple lobes different in either side of the lungs. You have gut is always towards the left. Then liver is always towards the right. All the things are arranged properly. But in a small percentage of the population, they have a mirror arrangement. It is like totally reverse. The internal organs are in reverse order. Actually, surprisingly, in both of these cases, people will make up to their full like life expectancy, like 75 years. So what happens if if the organs are jumbled up, like you can get congenital heart defects and several complications, and these patients suffer throughout their life. They will not make it up to 75 years. So the question is how this precise positioning of the organs are determined. We have shown that zebrafish is a nice model to understand how the symmetry break is initiated and amplified during development. Similar to humans, zebrafish has a functional heart in 24 hours and develops heart and gut in the first 72 hours, they loop properly. So we will be able to visualize all those things. So the next thing comes, like, what are the key signals that determines that? So embryonic left-right asymmetry happens during somatogenesis, the formation of somites. When there is an asymmetric flow generated in the left-right organizer, which is common in mouse, zebrafish, or in frogs, even in humans. And this asymmetric flow generated hits on the walls of left of the left-right organizer and creates differential signals. And the intensity of these differential signals will determine where the organs are placed in the body. So this mechanism is conserved in vertebrates, including humans, mouse, frogs, and zebrafish. So when you take zebrafish, Kupffer vesicle is the organ of laterality in zebrafish. So there are like these small projections or cilia in the chamber, and they create an asymmetric flow. That is how you can get the organ positioning in the zebrafish. Actually, you can visualize live movement of this asymmetric flow in the embryos. So we have a good model to study this problem. Now, which protein would you select? That's the next thing. So we have selected one of the myosin, Myo1D. And this motor protein is important for moving cellular cargo across the cells. Myo1D is one of the proteins used by amoeba to perform cellular transport and cell shape changes and motility. So we have a working model and we have a protein of interest. What will you do next? We can delete the protein from zebrafish and study what happens to those embryos. So we track the development of Kupffer vesicle during the formation of lumen. In the 4 hours time period, wild type embryos expand the lumen in the Kupffer vesicle, but mutants fail to form the lumen in a similar fashion to that of wild type. So to understand this clearly, we live track the vacuole movement in the cells. You can see the movement of these vacuoles towards the periphery of the cell. So this we have done by super resolution imaging. So you need to see that, like, we are actually tracking the embryo at a cellular level. and you are tracking the subcellular structures in that live. So in a mutant, the vacuoles are not able to move because they lack the motor protein to drive that to the surface near the lumen. So as a result, what happens? This is a time-lapse image of the video, and we quantified the vacuole trafficking speed. In the mutants, vacuoles are stuck in the cells, so the lumen formation is affected. So the long story short, myosin 1D requires vacuole trafficking in the left-right organizer for lumen formation. And when you lose myo1D, it can cause left-right patterning defects. So not only in the left-right organizer, this affects the other lumen forming structures like ear, aortic vesicle in zebrafish. So like many other papers, I was very proud of this work. But if you think zebrafish as a model for preclinical drug development, you need other supporting models. So I switched my gears like in the last 3 years and started working on another interesting clinically relevant disease model. So this is organ-on-chip systems. So here we are actually microengineering and recreating human organs in a chip, like outside the body. So we can use them to test the drugs which are useful in the clinic. In fact, FDA is promoting researchers to use 3D organ models to test the drugs in the future. So now you know what to expect when you get into this field. In the US, biomedical research is a well-respected profession because US success is like in developing a therapeutic drug will add value to the US economy. And also you are making a positive impact in people's lives. So the major driver or motivation for pursuing the career would be like your intrinsic motivation. Like you say that I want to pursue this research due to passion, curiosity, or purpose. But passion alone, however, will not hold you into that career unless you are rewarded with your efforts. For your career success, actually, there should be more of intrinsic motivation, although it should be complemented with rewards. So if you think about what you need what are the essential traits for you to become a successful biomedical researcher. So if you have nice observational skills, if you can manage multiple projects, if you are better in data analytics skills, if you can communicate your findings and make it you can make it a better story. If you are persistent in what you do, then, and like if you try to enjoy your journey, then this would be a nice profession to be on. Let me tell you, I did my studies when like there was land phones instead of iPhone, and we had like desktop computers instead of like MacBooks. But in the current era, we have like huge scientific advancement, like gene therapy capabilities, where we can create CRISPR babies of our choice. We can bioprint organ of our choice to transplant in humans. We have augmented reality, artificial intelligence, blockchain to enhance virtual capabilities. Then with all this, preparing for the competition needs new added skill sets. If you think about the current situation, it also gives new possibilities. So better be aware of the current market possibilities and then select a career that fits into your interest. So thank you so much for this platform to share my experience. And you can connect with me through email or LinkedIn if you wish to do so. I'm happy to take any questions now. I will stop sharing.
Thanks, Manush. It was a great talk, and I like how you structured your presentation. So for those who came in late, Manush just went through showing, what were the steps he took for getting into biomedical research area. So the steps from his PhD time in India and how he what's the pathway he used to get into an international fellowship, postdoctoral fellowship, and how he reached so far. And also he went through in detail what you can expect in a career path of biomedical research. So thanks very much for that. I have put a link in the chat box. If all of you can just spend 1 minute to provide a feedback, and also I can pass that feedback to Manush as he will be expecting for that. And if any of you are interested to join our coordinator network, please let one of the board members know. If you are interested, please send us an email at 2afstalks@gmail.com. So we're going to start our discussion. If you have any questions, please put them in the chat box, but also use the hand raise feature. And as of now, there's no hand raises, but if you have a question, you can unmute and ask. I'll give you 1 minute to think about the questions. So meanwhile, I put a couple of questions from my side, Manush, if you're okay.
Yeah, sure, go ahead.
I don't have any much complicated questions for you, but you're showing that drug discovery process, the the chain of development. So I was just wondering, I would assume towards the end of that cycle, it will be mostly the companies will be working on that thing. But what will be the up to what level in that process? As an academic research, you will be contributing. I assume there will be nearly zero towards the end of that cycle.
So, like up to mouse clinical testing.
Yeah.
Like I was early screening process, like development of candidate drugs, and you prove, like you identify the mechanism of action. Then you also test it in multiple models, like early, as I was showing, like it could be C. elegans, zebrafish, or even two-dimensional models. And you can directly, some of the labs actually takes two-dimensional screening. After two-dimensional screening, they take it to mouse and prove that it works.
Mm-hmm.
So that would be sufficient enough to tell the companies to take it for further development. So most of the time, like small startups bring it up to a stage where like big companies like Pfizer is like a huge biotech company where, pharma company where they can take up any projects and any type of clinical research. They have like huge funds. They don't need outside support. They can take up all these things and bring it up to the clinic.
Yeah, thanks. My second question is on a different dimension. As a person chasing for, a transition into biomedical researcher. So, so you were using zebrafish models when you were more on the fish side, and then at some stage when you went into a postdoc position which was entirely in biomedical research. So how did you feel as a person from a different discipline and applying for that position, and what was your challenges, because you have to probably compete with other candidates who are purely from a biomedical research. What was your experience?
So I'll tell you something, like, well, we have to be open and flexible. So that is the main thing if you decide to switch your career. So first of all, the techniques you use, whether it is in studying zebrafish biology or in a like a pure basic biomedical research in like cell-based assay or anything, the techniques would be similar. So you can apply all those transferable skills to gain experience in the new field. So that's how you make the transition.
Well, what I was just trying to visualize what sort of how would the board would have been assessing you, and what sort of questions you have faced in that sort of application.
You mean to say, like, when I get into a biomedical research from a fisheries background?
Yeah, if you're getting into a lab where there's no one have a fish background, but then you are coming from a different discipline, and of course when someone in that perspective is evaluating or assessing your application, and they will be expecting some sort of language in their world when you. So I was trying to understand how you cope with that.
Let me tell you, like, when you apply for a position which is different model system from where you have experience on. So if you work on a single model system, you will be an expert in that. You will be expert in only in that. So that is the problem. But if you are adaptable, if you are flexible, then if you are open to learn new techniques, new models, then you have opportunity. And when you get interviewed, they will assess all these things, like what is your attitude. People look for attitude, because techniques can be taught, attitudes cannot. So it is the attitude only, it matters.
Yeah, that was great tip. I'm pretty sure many of the final students probably won't think attitude as a big factor, because probably that is not a process. What no one has probably spoken. Because you would think evaluations are based on degree certificates. Maybe to the next level, yes, what techniques you have used, but probably not at the level in how compatible you are with other members in the lab. How would you approach a problem and everything. Yeah, thanks. If anyone have a question, please, you can ask directly to Manush. You can unmute yourself.
Hello.
Hi.
Hello, Manush.
Hi, how are you?
I'm not asking any question.
Okay.
It was a very nice and informative talk. For
Thank
me,
you.
Regarding this zebrafish, I heard about zebrafish using for this biomedical research, but now I understood how it is being done and how it is used. So it was a very nice talk.
Thank you.
That is what I want to say. Anyway, thank you, Manush, for such a nice talk. Thank you.
Anyone have a question, please, you can ask. And we can ask, I think, pretty much anything. I don't think you have to be, you don't need to have any hesitation, because if you have any questions regarding, how, any questions in regard to how, if you are someone interested to go abroad for higher studies, and this is an opportunity for you to ask any questions to Manush, even if you're not looking for biomedical research, there's still many things that Manush can actually advise.
Yeah, it can be two things. Either they understood everything or understood nothing.
Maybe not too much in the science part, but yeah, I understood the skeleton of it. Well, I have one question in that last part when you are talking about asymmetric herd case, that kind of thing. Like, why did you choose myosin? And is that the only one reason of getting to that asymmetrical stage? Or there could be other things, right, as well?
Like 1,000 genes.
Yeah. So how did you end up in picking that one particular one?
Yeah, because when we start a research, actually, we get into review of literature, right? So we look at like which protein to choose and what you have to go forward. During that time, 2 papers came in Nature about using one of the primitive model systems where it has shown to cause like gonad looping or gut looping by this unconventional myosin 1D. So that is where the start is. So like when you think about that, you need to, like, if you decide on pursuing something, before doing that, do thorough research. What is out there? You need to be the master in that protein, like about knowing all the aspects about that protein. So
I think this is another tip from Manush. If in having a research career, and how important is that you have to be on top of everything, and especially reviewing papers can help. This is an example. Like, you can get an idea by reviewing papers, but then you also have to put some dedicated time for it. So it's a balance of how much time you have and how you use it efficiently.
So I'll thing like you cannot do a research for making it to a Nature paper. You can never do that. But how your research is progressing, how that story is being made, how it is made relevant, that is what it takes a paper to a very small journal versus a Nature journal. Because the same story can be in a very low impact journal. So the relevance of this was like, we have taken a protein from amoeba, like, which is used for a particular purpose in amoeba, and we have shown it has an effect in zebrafish. So we have rescued the looping defects using amoeba myosin. So that is how it went into Nature.
So in some ways you are telling, it's not an unreachable.
Yeah, it is. It's possible.
Any questions? Manish, I remember you have been talking to me about the MEX, GSPS when I was in CIFE. These scholarships are still running, right? They are still on.
Yeah.
Yeah.
Now I think they have increased the fellowship amount, then all those things are there. But yeah. So if there is any like application process changes, which I don't know.
Yeah. Manish, can you I think there's no questions here, but I will ask for everyone. Can you give a quick guideline on how to approach the researchers abroad to getting into a lab, because most often you see that you send millions of emails to many people every day, and you get maybe 1% of them will respond to you and probably to say no. So do you think there's probably a lack of education on how you would write an email, how to approach and how to make you look different when those processes have millions of emails every day to go through?
Okay. So I would tell you like how to like get this positive connections. Like, first of all, before contacting a professor, you need to know his profile very well. And you need to relate that to what you're doing. Otherwise, they won't be interested in what you are doing. So the PIs always look for people who can bring new dimension, new value to their research. So definitely, like how you can relate your research or your work and your experience, your skill sets, how you can add value to their group. You have to highlight that in the covering letter. So you tell them in first 2 lines what you're doing. Then you tell them like how that research how that skill sets will help that PI or the group in taking forward their research. So I think I have a new message. I don't know what it is. How
your MBA helped you in your biomedical career by Sajana?
Okay, that's a good question. So,
yet to see the results, isn't it?
It's an open question. I just completed MBA. So I have listed all the opportunities in one of the slides. You have seen that PhD and MBA, you can limit yourself as a scientist, but having an MBA, additional MBA, will first of all improve your soft skills, like your perspective, your way of thinking, like the future potential, future possibilities. And second, in terms of opportunities, it will broaden your horizon to look for different opportunities. I don't want to tell that I'm going to be a single career in the next 5 years. But right now, even I cannot predict what it will be. But you open more possibilities. That's all. I mean, that's a vague answer, but that's all I can say.
It's okay. I can bring you after 2 years to give a proper reply for that. Manish, you talked about the attitude, how people look about your attitude. Can you just give a few examples? What does a good attitude look like, and what is a not so good?
So getting
This
well.
is for the final year students, because sometimes it is not that the line is not very clear,
Yeah. Attitude in the sense, like when you are in a team, you need to be a good team worker. You need to gel with your team members in the group. So that is very important, because no single person can achieve a really big goal. Like if you take up a huge research project and several people are working on that, and if others find you that hard to work with, then it's not a good way of going forward. So you need to work really well. That's what I was saying. Like, I mean, I am glad that I worked in biochemistry lab. So there are like 10 or 12 people working in the lab in the same goal. Everyone needs to get papers. And somebody will be good in writing papers. Somebody will be good in like looking at the data and analyzing. Somebody will be good at different aspects. But all were working with the same goal, and that helps. I mean, that kind of a training is really useful.
So that also emphasizes, how much important is to have research papers to get into positions abroad. And sometimes I think, most students I have seen who are looking for a position abroad, they don't have any papers in hand. But how would the, people abroad would see this? I got my PhD position abroad without having a paper in hand. But they did ask me they did ask me whether I have papers, and my answer was yes. I'm currently writing that paper, or probably I'm not very sure. Probably I said it's under review, and it was true though. But the fact that was that I didn't have one to show at that moment. But yeah, my answer was positive, and I did publish that later. But do you think that having a couple of papers will keep bring you well ahead of other applicants, right?
Definitely yes, because having more papers is good, and it does not matter. You need to have a high impact paper, but in your field, whichever field you are, and if you have like at least 1 or 2 papers to show, so yeah, that is better.
But the story of the academic research, you used to say, publish or perish. You have to keep publishing papers to keep your progression in that line of career. So how true is that, Manoj?
So you're telling me. Okay. So,
I mean, not everyone will be capable of doing that, keeping on publishing on papers, because it's sometimes not just your job. As you said, it would be, if you have a good team with you, it may be possible, but otherwise it would be a bit struggling.
Yeah. For example, like when you are in masters, like you need to decide about your goal, like whether you want to pursue as a student. As a student means you need to focus on GRE TOEFL and do that. And even when you apply for masters, and if you have a paper, that adds value in your CV to get the admission. And yeah, that's what I wanted to say. And depends, like you can try in different directions, but focus on early. That's better.
There's a question in the chat box by Oppo F1S. It's a phone name anyway. How can BFSC graduate or a fresher can apply for a job abroad? That's a very general question, though.
But that's a different topic from what my experience is. Yeah, I got out of the like for opportunities abroad, like very after doing a master's and PhD, and after that. So I know that there are other people have done after BFSC, like BFSC. With the BFSC also, since it is a 4-year degree, if you have GRE and TOEFL, you would be able to apply for masters. There are some successes like that. I think one of your batchmate, right, Deepak, you has done that.
Yeah.
Then yeah, so that way you can go abroad for masters. But if you apply for jobs directly, you need to have an H1 sponsor in the US. It's very hard to get. So directly, I doubt. But mostly people do that. I know one person have done that before. Just after BFSC, he went and worked in a farm in US. I think salmon farm or something like that. That was like through family sponsorship. So somebody has to sponsor your visa. Like if your family member is a green card holder or a citizen in US, then you will get sponsorship, and then you would be able to work in US fish farms or hatcheries or something like that. So that is the only way I know.
So my experience, after going to Ireland for my PhD, and having many colleagues who is doing a similar, higher study in Europe, I think the requirement of, TOEFL or GRE is specific for US, I guess. So TOEFL I seen that it's not very demanding. That they did ask me after joining for it, and I provided, but it was not mandatory. So I think it's not that strict compared to US, maybe possibly. So my advice is just don't worry too much. Give it a try. And even if you don't have a TOEFL, you still have a because the fact is that you are still having an education in English language. All your education, maybe from your degree, is in English, and so I think that was something they considered for my application and for everyone who came at that time. So that is something maybe that information can be helpful. But I know for US, GRE is a must, and there is a screening process, and we cannot get around that. So now we have some questions here. Now people have started asking questions. But just connecting with the previous one, there are different pathways to go abroad. And I will tell you the most easiest one is to get a spouse visa.
Okay.
So, Manoj, I have one point.
Okay.
Suppose one person got admitted in a lab, and that research started. By somehow beyond that capability or control of that person, that research not attain the target, what will happen?
Okay.
Any cases for such a situation?
Yeah, so many cases.
And what is the fate of that candidate?
I mean, I was telling about focusing on multitasking, right? Multiple projects.
Mm-hmm.
So you can show productivity in one of them, even though all the other research were failure. So they will be okay with that. First thing. And if all the projects fail, you may need to leave the lab because they will not keep funding. So there is in US actually, there is no secured job. Everything is based on projects and the success of the project. So there is no government or any job. It is not 100% sure that if you enter in that job, you will be working in that job for 60 years or 70 years.
No,
So
that what happened to that already that funds given to that candidate? Any he has to refund or not?
So the thing is, if he's coming up with a fund, then until that fund is over, then it is okay. For example, I'll tell you, like for JSPS, I was funded for 24 months.
Mm-hmm.
So until the 24 months are over, that is fine. But if the success of the project is not there, and if we are not producing results, we will have review like every 3 months, 6 months to see how this is progressing. So that's how it goes.
Any that they will get any extension or not?
I doubt. I mean, like in academic setup, mostly, yeah, it works. If you are in good terms with the lab senior PI, yeah.
Okay. Thank you, Manush.
I'll tell you an example right now. Like Google CEO recently told about this aspect, particularly this aspect, that you should not look at the result. You should look at the kind of effort your employee is putting in getting that project. That's more important than the actual output or the positive impact. So that is not the case so far, but that is a change in attitude right now coming up. I think I was talking to Sundar Pichai.
Thank you, Manush.
Yeah. So I think there are some questions here.
Yeah.
What is that?
So one of the question is how to start preparing for GRE and TOEFL for PhD admission in UK or USA, especially after MFSC. Is it tough to clear the examination? What's your take?
Yeah, well, I mean, I have taken this exam. It is tough. TOEFL is less tough than GRE, I can say. GRE, you need to like some of the books we normally use to. I mean, if we are not thorough with the vocabulary, that's the problem. If you are not good with the analytical skills and taking math, solving the problems, that's a problem. I think you need to put effort then and do your best. That's all I can say. Like there is no shortcut for that.
Sumant Kumar Reddy is asking, how can we know that whether assistantships are available in a project? And also he asks, Canada or USA, which is better for long-term settlement? The more practical questions, Manush. These are more practical.
So first question, I can say that like assistantships available in a project. You need to like, they may not advertise that all the time. They may not be up to date in telling that the assistantships are available. But you can directly contact a professor which is working in similar areas where you are working on. For example, I was working on stress physiology in India. So I will naturally look at for the people who are in the similar research area, who have similar interest. So they would be interested in responding to my emails. That's how I connect. Now, when I was doing this like search, there was no LinkedIn. But now LinkedIn is a very nice way to connect with the people. So you can use that. And for the second question, Canada or US is better for long-term settlement. Canada, I know that there is less people and they are promoting immigration right now. So, I mean, Indian community is getting really big, especially the Punjabis. And yeah, I think, so I mean, Canada is a good destination, I can say. But in terms of biomedical research, so the funding for biomedical research, top funding in the world is in US. Right now, China also is putting up a lot of funding for biomedical research. That's why it is progressing really well. So far, until now, US is one of the top destinations for biomedical research. Then long-term settlement, and if you happen to come to US, research stream is the shortest way you can get green card. If you compare with IT or any other jobs.
The other question is, what are the other fish species that can be used as an excellent model organism as a zebrafish in biomedical research?
There are other models, but none of them are so advanced and so not so commonly used. You have a giant danio, that is another model. It is also a zebrafish model, similar to zebrafish, but there is I'm actually blinding on the other species now. I think I don't remember. There was another model which I was exposed in Japan, but I don't remember now. Yeah, giant danio was one of them. Choosing what department in masters do you think offer greater opportunity to? Yeah, of course. That's a good question. That's a really nice question. Okay. So if you happen to be in nutrition, biochemistry, microbiology and pathology, or genetics and biotechnology, those are the best. And yeah, if there, I'm not telling that other fields are not good, because if you happen to be in resource management and if you develop like good data analytics skills, so you can secure postdoctoral positions in economics. So that's not a it's not anything different than what I'm talking, but it's in a different field.
Yeah, I think all fields have, if you are really into it, there are opportunities. But I think the question was more, general thing, like which ones have greater opportunities. So more opportunities maybe.
When you are doing something, do it with full dedication. That's all it matters. You don't worry about, oh, I am in this field, I cannot do that. Do good. That's it.
So I think there's no more questions. Maybe, oh, there's one more. So Sumant is planning to do a master's in USA. So he's asking, is there any positions available at present under you, Manush?
It's a open platform. I cannot declare.
Or maybe Manush, just put your email address. So if anyone has questions, they can send you.
Yeah, I'll tell you that like I cannot like hire someone right now, or I mean, previously I helped one of my junior to secure a position, postdoc position in US. I can say that. But if I cannot hire someone, I can help people to connect to a person where they can hire. So that is the way it goes.
Yeah, or you can just call it networking.
Yes.
The networking is very important, I think, because most people get jobs through someone they know, and people also hire in some ways if they know a candidate, because I think it's the compatibility with the candidate and other members in the lab is a top priority, even before what skills you have, what background, from what discipline you come from. So that's the email address. It's of Manush. It's in the chat box, manushsm@gmail.com. If you have any career related guidance questions, maybe I would say, you can feel free to ask to Manush. And he was one of the role model for me, and he have given a lot of advice for me when I was doing my master's. So I can definitely vouch for that. So thanks, Manush, for giving your time for this talk. And I hope this will be helpful for many other students and researchers who are looking to go abroad for getting higher skills or positions. So thanks once again, and thanks for everyone who joined today.
Thank you so much.
Yeah.